if you've checked with Scott, but that's okay. Great, thanks. So welcome everyone to the IICD monthly seminar. It's a great pleasure for me to introduce Scott Fraser, who's been a long-standing pal of mine. We were at USC for some time. Before he starts, I'm going to give just a brief overview of what he's been up to. So Scott had a long career at Caltech, which involved starting the Caltech Brain Imaging Center and helping to start the Kavli Institute for Nanoscience. But in 2012, he made the move across town to the University of Southern California, that's USC to us, and where he became one of the provost professors. And he's cross-appointed in the College of Letters, Arts and Sciences, CHLA, which is the Children's Hospital of Los Angeles, the Keck School of Medicine, and the Viterbi School of Engineering. This is because his expertise is in interdisciplinary research and translational science, and in particular imaging. He's, I think, currently got 63 patents, if I read the story correctly, and has co-founded 5 startup companies in recent years. He currently runs the Translational Imaging Center, the USC Bridge Institute, and he's Director of Science Initiatives for the whole of USC. So he has a stunning academic record and is a member of all sorts of things. I'll mention just a few. So he's the fellow of the National Academy of Medicine, the National Academy of Inventors, the American Institute of Medical and Biological Engineers, AAAS, and the European Academy of Science. So most recently, some of his work in developmental biology was honored with the Edwin Conklin Medal. So Scott's going to tell us something about eavesdropping on the dynamics of embryonic development. And it's a pleasure to have you here, Scott. The floor is yours.
Well, thank you very much. I'm really very tickled. I've been really enjoying your series because of all the tools that you've talked about. And what I'm going to try to do today is share with you some of those tools that we're developing. And I hope that this will share properly. So we will see if I can get this to do the right thing. So I think that's probably working. So one of the things we've been developing over the recent years is tools that let us, as invasively as little, follow developmental dynamics and follow disease progressions and follow the molecular and cellular interactions that underlie a lot of different developmental systems. And as Simon mentioned in the introduction, I've been very lucky to be supported by some amazing foundations and some fun programs at the national level. We've had a lot of patents that have been filed through Caltech and USC and the Alfred Mann Institute. And then we've got a few different startups and commercial relationships. I'll mention any of those if there's any thing where you need to doubt my veracity. But let me just sort of start out. But what we've been trying to do for quite a while is to work on this idea of systems biology in a relevant setting. By a relevant setting, I mean in an embryo where we can embody the cells and watch what's going on. And we love embryos, both because developmental biology is so fantastic, but also the events are relatively choreographed. And because of that, we can fit them underneath the stage of a microscope. And in the 21 hours or so later, we've got an embryo with a beating heart, with a working eye and a working brain. And of course, we'd really like to understand how that's how that's happening. Now, many different single cell analyses are being developed. I helped run the Cold Spring Harbor conference on single cell analyses, and I'm about ready to teach a course in Venice on single cell analyses. Of course, one of the really fun things is that you get a lot of data. One of the not so fun things is the variability and the heterogeneity. And what we've been working on over the last few years is to try to work on tools that let us look into embryos or into biopsies or into whatever tissue we might care about, and to use that variance as signal rather than something to curse at. And that's what I'm going to try to tell you about today. I'm going to draw, I guess, on the left side of the page today. The right side of the page just appeared, a paper just appeared in PNAS and a little write-up on it in Wired and a few other things on watching synaptic dynamics inside an animal while it learns. But what I'm going to talk about today, I think, is more the cellular things that are involved in molecular imaging and in intravital imaging of labeled and unlabeled tissues. And so I'm going to draw on this, I guess, in a little bit of a historic way, but I will mix up history a little along the way. One of the things that really has driven a lot of our work is trying to come up with techniques that let us do multiplexed detection. And multiplex detection for things like in situ hybridization has been a nightmare by the techniques we were trying at first, because the nonlinearity of the probe enzymes that are used to deposit chromogens, like you're seeing here with this fixed zebrafish embryo. I mean, the beauty is that it works. The bad thing is that it works in a very nonlinear way. For those of you that remember photography, it's a lot like printing on hard photographic paper. So if we have only a little bit of enzyme or a little bit of gene product or let it run for only a short amount of time, we get very little color. It saturates at the high end, and there's a somewhat linear domain in between the two. But this makes it a challenge to do quantitatively. And just to give you an idea, this sort of classic, single cell RNA-seq display with a log2 sort of color bars, in situs are great at seeing where things are, but not necessarily telling where they are and what amount. This is the same gene at the same stage in the same strain of mouse, and it happens to be in the same paper. And it's hard to say with certainty that those are the same patterns. So we've been working for quite some time to come up with a couple different approaches for getting quantitative estimates of transcript numbers. And that means getting a fairly good capture rate and a fairly good amplification. So this is a technique that we've developed first with Niles Pierce and his colleagues, and more recently have made an improved version that we're now pushing a little bit further. But the basic idea is that it involves using some of the concepts from DNA origami or DNA nanomachines, in this case based around hairpins. These hairpins would actually like to bind to one another, but they'd have to sacrifice all the base pairing that they have with themselves to find one another and base pair between the two. And so they're stuck in a metastable state. They're stuck there until an initiator is brought to bear. And the initiator, here I've just shown the sequences like as A star. The complement here in this toehold A allows it to bind, and then for every base pairing that's lost in the hairpin, the initiator can make a base pairing, and that opens up the hairpin and reveals what is the initiator for the second of these hairpin pairs. And so now what we can do is have this amplify until it gets to about a 200 mer. Now the little stars I show here are meant to be dye molecules, and so we've got spaced out dyes on a leash tethered together. And to make this into an in situ hybridization technique, or a technique that lets us stain anything, as it turns out, we use this initiator and different hairpins with different dyes. So they're chemically orthogonal. If I run this with blender extract of zebrafish, the hairpins run in this gel as this small molecular weight species, this gamish down here of 4 different hairpin pairs. If I add the red initiator, then the red disappears from down at the dye front and appears as this 200 mer. If I add the green initiator, I get a 200 mer of green, it disappears from down here. So this nice orthogonal behavior allows us to make it into an in situ hybridization technique by hooking up the initiator sequences to our antisense probes. Then what we can do is wash in the hairpins. And when we do that, we get a nice linear gain to our in situ hybridization. And we also get reagents that penetrate into the preparation very nicely. Because it's linear, things like the nascent transcripts that are largely invisible in a conventional in situ show up nicely. So if we focused up and down, you'd see that there were 2 dots inside of each nucleus in this somites of the muscles of the developing zebrafish here. And as we go deeper into the embryo, in our work and in work that the Disseroth lab did to validate the tools, basically we get the same intensity independent of the depth. So this allows us, and I mentioned earlier that it's linear. 2 key things, if we're thinking about it quantitatively. 0 is not where all the intensity ends. The intensity starts above it. Then I don't have to have divine inspiration to know what's signal and what's background. And you can see if we recognize the same messages with 2 different colors, the plots of the colors are nice and linear and pretty nicely clustered, and we don't get the big fishtail that you do sometimes when they've got a low capture efficiency technology. So this has allowed us then to do a number of different biological systems, but it's also allowed us to do a lot of biological systems where we're living in this pyramid of frustration. What we really want is an infinitely resolved image captured immediately throughout the depth of a specimen. And the problem is that all of these goals are opposed to one another. So if we try to increase the resolution, we hurt ourselves in speed and depth almost always. So what I'm going to try to tell you about today is some of our efforts to live more happily on the pyramid of frustration. One of those is to make the labels brighter and to make them faster staining and restaining. And this involves a version of HCR that we've developed that we call Muse. Muse uses smaller hairpins that amplify more rapidly and separates out the labeling a little bit differently. So we get the same sort of polymers being formed wherever we have the initiators, and that can be connected to an antibody or an in situ probe. And then we can read them out by putting in labels that we can see in the fluorescence microscope or in the CyTOF or in the NMR or you name it. So it's multimodal, which is what the M in Muse stands for. And it's allowed us to get another order of magnitude or 2 over what we are getting out of HCR. So here in these stained Drosophila embryos, you can see that a standard Muse stain is about 10 times as bright as HCR. If we do another round of gain in that, we get a linear gain, but it's about almost 200-fold brighter. If we hook up quantum dots, which work relatively nicely, we can get up to about a 200-fold brighter in situ. So what that's allowed us to do is to get brighter and better contrast in situs. And here we're recognizing the same messages in some brain tissue by HCR and Muse. When I equalize the intensities, as I've shown here to the right, what you can see is that the Muse gives us both brighter signal and better contrast. So it's a lot easier for us to do automated recognition of what's happening in the specimen. Because we can hook up these initiators to a variety of different primary antibodies, we can do 4-plex or more with the same species. For example, this is rabbit antibodies you can see to the right. We can do in situs, and we can do a mix of in situs and immunofluorescence. And I can show people at the end, if you're interested, it works really nicely in the CyTOF and gets us enough gain to where things that you can't see in the CyTOF, we can see reliably. So this is a dated slide now, I realize, because it's about an order of magnitude and a half smaller than what Long Kai's used it for in his serial seekfish sort of approaches for looking at staining. So the good news is this works. The bad news is that it's 5 gigavoxels in that mouse embryo. And 5 gigavoxels takes a while to image because 10 to the 9th times any amount of time is a significant amount of time. And it also involves some tiling, so you can probably see some bleaching here. So we start to be nonlinear because of the imaging itself. And of course, what we really want to do is go in and count the messages, and that would slow us down 100-fold or so more. So that'd be 1,000 hours of imaging per specimen if we did it just blindly by trying to scale up. And that's clearly not going to be practical. We also want to be able to do more and more colors per round of staining. And so I'm going to try to answer both of those very briefly and show you the ways that we're trying to deal with this pyramid of frustration by using different imaging modalities and different processing. I should say first that all of our efforts to make faster and faster confocals, we are involved in both of these efforts, trying to help companies. And in one case, well, I guess in both cases, the instruments were commercialized. They end up generating faster images by using resonance scanners or slit scanners. The problem is that they bleach faster as well. And so it's a real trade-off that we don't want to make. They always trade off that bleaching for the speed, because while we're imaging one plane of the specimen, we're bleaching above and below. And of course, in an image stack, we're multiplying that up. Now, we were some of the early adopters of 2-photon microscopy to get around that excitation above and below. You can see here a dye cuvette and a microscope objective. With 2-photon fluorescence, you're not getting the excitation above and below. That's great news. It lets us look into specimens and see things. For example, Willie Zapato's image to the left, a former postdoc, looking at the brush border in the intestine. Some of Peter Friedel's work looking at tumor and tumor invasion. They're fantastic what you can see, but if we're trying to image faster, it's not a way to go. It's slower imaging. So what we need is something that goes deeper and faster. Because faster is Lightsheet. Lightsheet allows us to jump onto the bandwagon of Moore's Law. Moore's Law has made cameras better, cheaper, and faster, and lower noise at an amazing clip. And so if we could excite a whole optical section at a time and collect the image in parallel, we can take advantage of that. Of course, this is a technique, Lightsheet Imaging, that's getting reinvented, I'd say, every decade for the last century. It won some chemists a Nobel Prize back early in the 1900s, neuroscientists in the late 1900s, and then developmental biologists in the early 2000s. The problem with this is that blue light is pretty well scattered, and so we can't go as deep as we'd like. Now, 2-photon can go deep, but is slow, and Lightsheet is fast, but has this light scattering. So what we've done is gone over to a 2-photon Lightsheet using an infrared pulsed laser to shoot from left to right here. And what you can see is as the photons are not yet concentrated, there's no excitation. We can use that to shine through an animal's skull or through its bone, for example, to see the bone marrow inside. Then we can get an excited sheet here of, in this case, labeled beads inside of a scattering media. And then as the photons get less concentrated, the excitation goes away. So this has allowed us to pose this laser sheet in a number of challenging settings, and allowed us to see things as simple as developing Drosophila embryos and show that they develop at their normal rate and with no perturbation at all, and hatch just exactly on their normal timetable. And it also, though, has allowed us to do things like image brain function. In this case, these are some images of looking at behaving brains over time. This is part of a 2-day-long time-lapse movie. And the intensity at the end and the intensity at the beginning was basically the same. So the little bit of synthesis of new label was enough to keep up with any bleaching that we were doing at collecting these 2 seconds per volume brain images. And so what this 2-photon light sheet has done for us is allowed us to make good on the 2 photon advantages and justifies the $100,000 or $200,000 we spent on the lasers, but collect the images maybe 2 orders of magnitude faster than we could with a 2 photon point scanning system. It allows us to go faster and faster, and I'll show you some of the imaging we're doing later, but I'd say by a year's end, we'll be able to do hyperspectral 2 photon light sheet at maybe 16 megapixels. And that starts to be game changing if we can do that with high efficiency, and it looks like we can now. So that's one way to solve our problem of going faster. We can get down to minutes per volume instead of hours per volume, and maybe even seconds per volume if we're lucky. So what about the problem of the overlapping spectra? And the problem of the overlapping spectra is really significant because any of the fluorochromes that we care to use, even though we call them something like green fluorescent protein and yellow fluorescent protein, overlap more than they are separated. And that means that if we look at the spectra of multiple different fluorochromes inside of a specimen, even if we use really specific filters to try to see only the GFP, you can see that we let lots of things other than GFP through this bandpass. And of course, as we make this narrower and narrower, we're going to make it reject more and more of the emitted light, so we get less and less efficient. Years ago, we started working on this by building spectrometers into our microscopes. That was a powerful way to borrow from the remote sensing community and be able to take a few different spectral bands and the known spectra, for example, GFP and fluorescein, and to reliably separate them out. So you can see the GFP H2B and the fluorescein phalloidin being nicely separated. But if you also were eagle-eyed, you'd see one of the main problems with the linear unmixing we were using. And that is in the middle of this red field, the noise behind the green nucleus that's from the limited number of photons that nucleus is giving up gets imposed on the red label, even though the red label is quite a bit brighter. This ends up, there's this noise propagation limiting the utility of the unmixing and limiting the number of labels, because we're always limited in the signal to noise by the worst of the channels. And this was the situation we lived with for quite a while until Enrico Grattan and 2 of his amazing grad students decided to join the lab as postdocs. And what they have been using was a trick called the Fraser transform, taking a spectrum like the green spectrum that would come down here from this region that's green fluorescent protein, taking a Fourier transform and plotting the sine and cosine term of one of the harmonics, it becomes a green dot. The red spectrum becomes a red dot. And because we're in this Fourier plane, it turns out that a 50-50 mix is a dot halfway in between. It turns an inverse problem into a forward problem. It makes it tremendously faster and better performing here in separating out green fluorescent protein from yellow fluorescent protein. We now have this working in a couple milliseconds instead of a couple seconds. So really at real time, able to keep up. And the basic thing that helps us here is that the normal approach for collecting 2 photon or, well, any hyperspectral data is to take the emission, put it through a detector that separates it into 32 channels or so. That's great, except it now drops the signal per channel a bit, but it allows us to collect the whole emitted spectrum of what's in that specimen there. But now we've got to deal with the 10 to the 7th or 10 to the 9th different voxels that were in that, and we have to do some matrix inversions on a very large data set. By using the hyperspectral Fraser approach, we're able to Fraser encode. That Fraser encode involves dropping these 10 to the 7th elements down to maybe a few 100,000 elements at most, maybe even fewer. We can spectrally denoise them within the Fraser plane without changing the spatial arrangement. And if we think about this as a series of histograms, what we end up with now is histograms that we can unmix from one another after this denoising. And what was 10 to the 7th calculations becomes maybe 10,000 or maybe even fewer. This is why we end up being so much faster. We've compressed it by going with the Fraser encoding. We compressed it further by doing the unmixing on the Fraser plane and then using that in the display. And that allows us in seconds, and as I mentioned, we now are getting data like this out of a GPU in milliseconds to separate out multiple different fluorescent proteins. And even with only a few channels collected, we can separate quite a few different labels. And what's great for us is instead of having to use 4 or 5 different lasers to excite 4 or 5 different dyes, for example, in this case, all but one of them was excited with the same laser, even though it's far away from the maximum for, say, 458 laser working on citrine. So this has really changed what we can do around the lab, but we still had a problem. And the problem is what I want to do is eavesdrop on things as they're going on. And that means we want to be able to look at this with a minimum number of photons. We want to look at the lowest signal and lowest signal to noise we can get away with. And that means that a beautiful spectrum, once we look at only a handful of photons per label, can really drop down to something where the stochastics end up winning. This is where we've been able to use UNet and other machine learning approaches to try to restore the spectrum. This shows one of the training sets, and we've been able to train this very nicely using very low signal to noise data and ground truth data collected with many more photons, or even to use synthetic data where we've exactly modeled the behavior of the microscope and get a series of predicted spectra out that are good enough for us to reconstitute images that would have otherwise had significant stochastics contributing to bleed through and the inability to see cell boundaries, for example. Once we're using this machine learning enhanced version of the linear unmixing, we can see the cell boundaries pretty clearly. And so this has really changed, again, maybe another order of magnitude in the light that we're able to use. And it means we can go dramatically faster, see dramatically deeper, and deal with high dynamic range data, things where we've got some things that are very bright and some that are way less bright. That has allowed us to very accurately take the autofluorescence from the specimen and to separate it into some of its different components. For example, some cells that fluoresce, retinoids that fluoresce, and then NADH free and bound, which also fluoresces. This has allowed us to think about a different sort of imaging. So far, everything I've been telling you about has been dead zebrafish or transgenic zebrafish. That's a good use, and we're really happy with it. But if we're thinking about clinical utility, or we're thinking about even using it in biopsies, very, very few humans, although probably more of our lab mates are transgenic with time, come with transgenes labeling the key players. So what we've been trying to do is to find out if we can use the fluorescence lifetime instead of the wavelength as a way to understand these things. In fact, what the instruments we're building now look at the lifetime as well as the color of the spectrum. Now, lifetime is the time it takes between a dye being excited and the time that it emits the photons. And that's normally seen as an exponential if it's a single component. Of course, as it gets richer, it starts to be this multi-exponential. And it's the same problem that we've had before with trying to see spectra when we're trying to unmix those. These become messy and ill-posed problems. This is where the Fraser transform from Enrico Grattan and his colleagues have really helped, because it takes what was a painful inverse problem and makes it into a forward. And for example, if it's a single exponential, it's going to be on this universal circle. So you can see T1 and T2 along the circle. Any mix in between the two of those two, or you could have a mix of three, will be the arithmetic mean of all of those. So this ability to use the Fraser approach for rendering and unmixing has been an amazing help to us. It means that we can look at things like the energetics of the cells, because within the cells, the NADH, when it's free, well, largely cells that are in glycolysis have their NADH free, and the free NADH can interact with the water around it, and so it has a very short lifetime. I've shown on the Fraser plot here where free NADH would be. All the different enzymes that are involved in oxphos can bind to that NADH. And all of those protect the NADH from the water. And so it lengthens the lifetime. So what we can do is look at a specimen and look at the lifetime of the NADH fluorescence and be able to read the amount of free versus bound NADH and read the metabolism of the cells as a result. And of course, because it's the Fraser and we can separate out other things, we can even look in tissues that have significant autofluorescence like this one here and be able to unmix that out as well. So if we sort of look at what Enrico Grattan and his colleagues have done, they've done some amazing jobs going into complex tissues like the intestine. And the autofluorescence in the intestine is just awful. But even in the presence of that autofluorescence, they can look in and in this, with this green label, be able to see the stem cells by their characteristic NADH state, as well as you could see with the transgenic and live animals, and well, in fact, probably better than you can see with the transgenic. And so he's been able to take advantage of the slightly different redox states of each of the transit amplifying cells and the stem cells to do imaging without the need for exogenous labels. To give you a feeling for this, I should say we've used this technique in the past. It's a technique we both love and hate because the technique is super powerful, but the equipment was super painful. We are very lucky that Leica has built a confocal microscope now that makes this a lot easier, and has recently, working together with us and some others, put the Fraser analysis inside of it. But it means that now somebody without having to train in applied physics and everything else that was involved in getting good film imaging can do things like take a movie of a mealtime for a cell culture dish. This is just adding new media to the culture dish. You can see this dramatic change in the metabolism of the cells. You can also see two heterogeneities in this. You can see cell-to-cell variability, and you can see intracellular variability, different regions of the cells that are different, even to a very subtle change, like just giving them new medium, as we're showing here. So this is where we decided to go after a cancer-related problem. And this cancer-related problem has been to look at what people often call the side population of cancer cells, sometimes in quotes, the cancer stem cells. So what's in a cell sorter, you can separate out by dye exclusion. In this case, it's a nuclear staining dye, a group of cells that doesn't stain, and then a group of cells that does stain. So that are called the non-side population, the mass of the cells. And these are relatively low tumorigenicity when they're implanted into mice. They're pretty susceptible to chemotherapy. They really seem to be the run-of-the-mill cancer cells. The side population, the one down here, are irksome because they're very tumorigenic, they're very drug resistant, and they have a lot of pluripotency genes expressed, hence the name cancer stem cells. Now, of course, once the cells are killed and run on the flow cytometer, it becomes a little hard to analyze this. But trying to do this in live situations, live sorts, and then following them over time people have been able to enrich in the side population cells and watch as those go away, and then watch as the fraction of side population cells come back, and then watch as they go away and watch as they come back. And this is one of those experiments where people have argued to the near death that the red cells at the end are the same cells that keep on re-emerging as the side population. They're just expressing that nature. Or where this is much more stochastic and the cells are creating some set-aside cells that are the side population. And so during this cycle that we're seeing in these mass cultures, the ones that are the side population at the end might not be the same cells as the beginning. So we got together with Amir Goldkorn to analyze these cells, and we've done this now for a number of different settings. Nick Graham and his colleagues did the metabolomics with mass spec. Others did the single cell RNA-seq. And what you can see in this plot and this well, anyway, is that the side population versus non-side population changes in its expression of OXPHOS versus glycolytic markers. It also changes in its expression of stem cell markers. And so the work is congruent, but at the 65 mil dish level of the gene enrichment for side population versus non-side population, the mass spec, the seahorse data, etc. Now, what we wanted to do is to get out of this situation where we're trying to infer what was going on and trying to interpret what dead cells would have done if they hadn't been killed and stained or their contents flown in a mass spec. And we decided to go after it using the FLIM imaging to try to understand the motion into and out of the side population. So first off, can we see the difference? I realize that this image may be a little too small to see the different colors, but I'm hoping, let's see if I can highlight this. I'm hoping you can see that that's largely green, and those are largely different colors from green. And so it allows us to say that the side population and the non-side population right after a sort have different lifetimes. You can see that to some extent at a population basis here in an enriched non-side population and an enriched side population. You can see that their lifetime histograms don't segregate out from one another quite a bit. But since this is a vital technique, it means that we can follow individual cells over time in normal conditions or in conditions where we've used chemotherapy agents or other perturbations. And this is just a plot. Well, here's a movie down at the bottom following cells over time, and we've color coded them in with their lifetime data, so we can watch as a cell goes from a non-side population cell through to being a side population cell. So pretty clearly, these are a distinct state of the cells, but not stem cells in the normal sense of the word. So we can see the transition both ways, and you can see that transition sort of schematized here, following a little bit more than a half dozen cells. A few of those become side population cells, and a few of the side population cells become non-side population cells in a time-lapse movie that lasts over a couple days, where we're following them every few minutes so that we're sure we're seeing the same cells. We also see big changes in their signatures in response to therapeutics. And we're still trying to parse this apart, but you can see how major this change is between untreated and treated cells. And we're still trying to understand, is this a selection? It looks like it's faster and larger than we'd guess if it was a selection. It looks like it's a strategy the cells are using to change their redox state in order to make themselves more resistant to a chemotherapeutic challenge. So we still have a lot to do on this experiment. We're still trying to carry it into experimental animals and into biopsies rather than patient-derived spheroids. But we're really having a lot of fun with it, and it'll be an enjoyable story where we're trying to make this technology accessible to the clinical researchers up at the School of Medicine using some of the tricks that we're developing, down in the engineering school. So I'm going to stop here and just sort of say that our whole goal here has been to add dimensions to our imaging. And of course, if it's a live imaging, we can add time. If we can use unmixing and do the multi-label sort of things, we can add colors and multiple gene markers and other things. Or if we're doing the in situ hybridization, although it's fixed, we can go to high multiplicity of labels. The spectral phaser analysis in the time that Enrico Grattan's alumni were in the lab, Francesco Cotrale and Mimo Arnesano, they were able to move the analysis time from minutes to milliseconds. And so it really has changed what we're able to go after. And what I hope I was trying to give you a flavor for at the end, and I hope it came across, is that we now, using this FLIM imaging and metabolism, can really start animating some of the results we are getting from the fixed specimen or in situ hybridization, or the single cell RNA-seq, which of course is a destructive technique, or the mass spec data or seahorse data that's been collected in more bulk cultures. So I'm going to end by thanking the people that made this possible, the folks that built the microscopes for the most part, Ty and his colleagues within the imaging center. The lifetime imaging was really pushed forward by Jason and working together in the past with Mimo, but now Mimo's moved on to greener pastures, and so Jason has taken that over. The hyperspectral processing has really been driven forward by Francesco Cutrale. Francesco's research faculty here at USC and just doing amazing things. And finally, the multiplex in situs I was showing you were largely driven by Simon and his colleagues. And I'm going to stop there and see if there's any questions or anything I can clarify with some of the other slides that I've got held in reserve.
Great.
Oh, and I should mention, before I do that, this is a baby picture, a product that we've been working on with Leica to try to make it so that a microscope can sit in a clinical lab or a research lab without its own dark room. The Leica Mika takes some of these spectral phaser things and is able to do single snapshot color imaging, multi-label imaging, and all of the life support and everything is built into the microscope or into its base. So it reduces the footprint. And so we've been having great fun using that around the lab, and hope to help them make it even better and more able to go after some of these things as time develops.
Wonderful. Thank you very much indeed, Scott. I'm sure you're up for some questions. Let's see.
Always.
What we can find. So any particular questions? I'm just looking for the chat thing. See if
I can answer one question that we almost always get, which, as we go to fewer channels, do we also have to go to fewer labels? And one of the nice things about the phaser transform is it's allowed us to disambiguate the labels pretty nicely, so that we can, out of only say 3 or 4 different channels, and we're building 4-channel phaser cameras now, be able to disambiguate multiple dyes apart. So it's a mistake to think of this as a 1 dye per channel sort of approach, the way we've done it in the past. Oh, and I think John Hunt has raised his hand.
This is a totally footnote technical question, but since no one else is jumping in, I'll go for it. When your hybridization amplification, you said it tends to limit at about 200, you said 200 base pairs or something like
Or
that.
200 hairpins
200
assembly.
hairpin, okay.
Yeah.
And so why does it limit at that point? Is that set by the equilibrium between the self-hybridization and the trans-hybridization at a given concentration level? Or why does that just stop? Or is that inaccuracies in the synthetic something?
Yeah, so
yeah.
well, I should say we've made it deterministic as well. We've made it so it makes a finite dendrimer and then stops. That takes an extra hairpin or two to make that happen. And we get slightly more reliable results. When the amplicons form, it's like some of the old stat mech problems that we used to torture the students with. We get to a point where they're long enough that that they reach a steady state in their gain. Even if we add a more excess hairpins, we tend not to get them much longer than that. It's possible that part of the limitation is the low level of poison pills, or whatever you want to call the synthesis errors that could be there within the hairpins. And we're now trying to do a series of experiments to really parse apart what it is that separates those, what it is that limits it. Is there really no limitation if we had pure reagents and it had pure access and it was a well-stirred reaction? And it will be interesting to see how it comes out. I have bet on both sides of this, and I've been shown wrong in both cases. So I guess that's almost impossible, but it means that the results are still in turmoil.
It does not seem to be simply the mass action effect of the concentration of, is there something more complicated than that? Yeah. Thanks.
I should say, though, that the good thing is that we've tuned the conditions to where the initiation probability, which is, of course, you could have fantastic gain, but if it only initiates on 5% of the messages, that would be a problem. The initiation probability is up in the 85–90% range. And so we think we're seeing as close to as many messages as there. And so that's one we're trying to really beat into the ground now, to understand in this technology and on others what the probability of capture or probability of detecting is. And right now it looks, as I mentioned, that we're at an 85% plus, and 2 other labs have gotten a similar number.
So this is just a nerdy follow-up to that. And so when you get down to the detection step of it, I mean, increasingly there is literature that mRNA contains covalent modifications of various kinds that may be involved in various things, degradation and or translation efficiency. Any sense how the hybridization efficiency, whether you can get at that in any way with that stuff?
So yeah, while we're nerding out, yes. So there's a lot of expected results. So you can add unlabeled probes or even just complementary sequences to help unwind secondary structure and increase capture efficiency, or increase capture kinetics at least. And because we can get enough gain out of even a single binding event, we have been looking at some of the probes that seem to be unreliable. And we think that some of those are recognizing post-transcriptional modifications or other ways that limit access. And so that's going to be very fun if we can use this as a way of following in the fixed cells. And we're now trying to come up with reagents that may even let us go after some of these same questions in live cells, using tricks from other labs like Rob Singer's and a variety of others that would allow us to take advantage of really doing the bookkeeping and dynamics of some of those modifications. Right now, most of those we're blind to, and most of the single cell RNA-seq, of course, is dealing with analyzing data with very low capture efficiency. And so that's especially blind to that. And so we're hoping that this is a niche that we could help follow those those post-transcriptional control mechanisms and look at regionalization within the cell and a variety of other things.
And just looking for any more raised hands.
Someone in the chat put a question.
Okay, from Nathalie. Not familiar with spectral. What is the load of processing on the image to get the data of interest? Can all imaging data do, can all spectral system do spectral imaging? Right now, we're trying to develop tools that do simultaneous spectral imaging. And that's because if a cell is moving, we'd want to be able to capture all the different wavelengths at the same time so that the movement or anything else doesn't limit it. And so we've been making high efficiency spectral cameras. We have one that's better than 85% that we're now trying to scale up to a high pixel count, as I mentioned earlier. And that's been allowing any photomicroscope to collect spectral data. Of course, many of the LSMs that are out there can collect, well, the Zeiss's, I think it's 32 channels, the Leicas, it's 5 channels. We've been able to do spectral unmixing on both of them. So you can put them into a mode in which you can get spectral data. And one of the things that's sort of most exciting is we're now trying to see how few detectors we could get away with. Because the temptation as physicists is to split up the signal into smaller and smaller bins. And of course, that means that we get fewer and fewer photons per bin, and the detector noise becomes more significant. The approach we're doing now is to use 4 detectors with a sine and cosine filter in front of them. This optically does the Fraser transform, basically. It's an approach that Enrico Gratton validated by taking 3 different exposures, and we've made it a version of this that can now take that in 1 exposure, and even counting the inefficiency of the camera that's doing the detection, we're at 85% photon efficiency. So that means that it's going to be a lot easier to add this to other systems soon. And my big motivation, and why the other reason I'm emphasizing the simultaneous nature, is that in most of our imaging, most of the time, and I mean 95%, was spent changing the filters rather than collecting the data. And so now we have devices where there's no filter changers, and that allows us to work at full speed. And that allows us now to think about voltage sensitive dyes in the nervous system and other things where the transients might last a millisecond. There's not enough time to change a filter and collect the data. So that was a very long answer to a short question. So my apologies, but it's been fun.
Great. So I can't see any more questions at the moment, but people have an opportunity to talk to Scott after this talk. So those who need to escape for any reason can do that. I think it's Lorenzo, it's on the same Zoom.
The same Zoom.
Okay.
Oh, okay, great. So it's just the same Zooms. I don't have a chance to get lost again.
No, you can do that as long as you come back.